Neurons, which are one of them is, well, many of them are pictured right here. You know what? Let's just label this right from the outset. This is a nerve. This whole thing right here is a nerve. And each one of those individual circles is a neuron. And over here, this is just an inset, and each one of these individual circles is a neuron. Neurons are highly specialized for their job, which is to conduct electricity. We also call that an action potential. So neurons are very long-lived in your body. They're one of the original cells. And thank goodness, because they do not like to do mitosis. So if a neighboring neural cell dies, its neighbor will not do mitosis to replace it. Neurons use a lot of energy. They need a lot of ATP, and their little mitochondria need a lot of glucose and oxygen to make it. And this is why, when you study, you have to eat. Neurons can be divided up into three areas. There is a receiving area, which is basically up here. This is receiving. This part of the cell is called the cell body. It contains the nucleus. This is the nucleus right here. And it also contains all of those other organelles that we know about, like an endoplasmic reticulum, a Golgi body, maybe some mitochondria. So it has all of that other inner workings that we learned about in chapter 3. In the case of a neuron, all of these little organelles work together to make neurotransmitters, which are the protein product of neurons. Specific to these neurons are these histological structures called Nissl bodies. And actually, Nissl bodies are just the endoplasmic reticulum taking up a very specific stain. Once made in the cell body, which can also be called the soma, I have heard it called the peri, which means around, karyon, which means chromosome. We're going to use cell body. But once a neurotransmitter is made in the cell body, the neurotransmitters are shipped in vesicles all the way down to these axon terminals where they're stored, right? So these are axon terminals. These axon terminals are like the sending portion of a neuron, the shipping portion. And it's here that an action potential in this neuron will turn into neurotransmitters to be given for another neuron. The axon itself is usually called the conducting region. It can also be called the propagation or propagating region. It can have a couple different names. These branches coming off the cell body, these branches are called dendrites. These are like little tiny antennae that are made for receiving signals from other neurons. So like there are axon terminals of other neurons here, right? And they're also releasing neurotransmitters. So the dendrites are able to receive those signals. On these dendrites are tons of ligand-gated channels. All of these channels, they open when neurotransmitters come from the other neuron. So this area between the receiving and sending portions of neurons is called the conducting or the propagation area, as I said. And this is the axon, which is similar to an electrical cord wrapped in insulation. We know that these little pillow-looking things here are myelin sheaths. There is an entire other mini lecture for the myelin sheaths, and they basically smother the axon. And this little bump right here is the nucleus of a Schwann cell, right? That's making those sheaths. And we know also that between the sheaths is called a node of Ranvier. And this is where there are ion channels that are exposed, whereas the ion channels under the myelin sheaths are smothered. Information or action potentials are supposed to flow from the cell body to the axon terminals. This type of movement is called anterograde. Action potentials, neurotransmitters, they're all meant to travel anterograde. If something moves in the other direction of movement, it is called retrograde. If you get bit by a rabid raccoon that you pulled out from the crawl space under your house, you could become infected with the rabies virus. What the virus will do is it will enter into neurons nearby to the location of the bite. And then those neurons, the virus, allow it to eventually make its way up those neurons into the brain by traveling retrograde in a motor neuron. Rabies is a central nervous system disease, and its most damaging effects are done in the brain and the spinal cord, but it has to get there somehow. And the way that it gets there is by traveling retrograde into the nervous system, into the brain and spinal cord. Neurons come in many shapes and sizes. The most common type of neuron is the one that I have here on the left, which is called a multipolar neuron. When in doubt, we are probably talking about multipolar neurons. I feel as though it looks like a tree with roots and branches. In fact, I will draw this many times just looking like this. I invite you to have your own picture of this. We will always use multipolar neurons as our example neuron. These types of neurons, they are the most common in the brain, and they're also the most common motor neurons in the peripheral nervous system. This next neuron is called a unipolar neuron. And in the peripheral nervous system, almost all of your sensory neurons are unipolar. In this class, we will always consider unipolar. The cell body of a unipolar neuron is like hanging off the middle of the axon, right? It's not at the end of the axon like the multipolar. It's hanging off the middle of the axon. Kind of weird. These last three neurons are called bipolar neurons. Now that you're familiar with the terms that describe the anatomy of a neuron, it's important for us to understand the types of membrane channels and their distribution in the membrane of the neuron. So leak channels, which I'm going to highlight in green because this is a leak channel right here in green. Leak channels are everywhere throughout the neuron's membrane, everywhere. Remember, leak channels allow sodium and potassium to leak according to their concentration gradient. And so in with the leak channels are also sodium-potassium pumps that return the leaked ions to their original place. Ligand-gated channels, which are these purple ones here, they are only located where this neuron would be coming into contact with the axon terminal of another neuron. And so for our purposes with this multipolar neuron, that would be where all of the dendrites are. All of these dendrites have ligand-gated channels because these ligand-gated channels are able to respond to neurotransmitters. These wouldn't be on the axon or the axon terminals because we don't respond to neurotransmitters there. We kind of know about that process from learning about the movement of acetylcholine across the synapse that results between a neuron and a skeletal muscle. But with neurons, there's a lot more going on. There are voltage, let me highlight ligand, but there are also voltage-gated channels that exist along the, and let's write voltage right here. Voltage-gated channels exist along the axon hillock, especially. They exist on the axon itself between the yellow myelin sheaths on this picture, and they extend down into the axon terminals, but not the entire way. These voltage-gated channels, they all depolarize in a coordinated fashion. If we can generate enough depolarization with these ligand-gated channels, then we can open up enough voltage-gated channels on the axon hillock, and we can get the action potential to hop, skip, and jump down this axon.